Namaskar dear students, welcome you to our channel by Gulab Sir. As a part of series of lectures, today's topic is protein. We already learned about carbohydrate, fats and DNA. Protein is another bio-macromolecule. It is a macromolecule because it has thousands of amino acids linked each other by peptide bond and form a very long polymer of amino acid called protein. Here it is very clearly seen that these are the amino acids. We learn about structure of amino acid and there are 22 total number of amino acids. Out of only 20 amino acids take part in protein synthesis, and protein is synthesized only and only in living cell with the help of the genetic makeup present in the nucleus on DNA. So proteins are synthesized with the information from DNA present inside the nucleus by the process of transcription and translation within the living organism. So whatever we eat protein, they are not directly used by our body. They are digested into amino acid. Then these amino acids absorb into the blood, through blood reach to the cell, and where these amino acids are linked each other in a sequence coded by DNA. It is not what as it is they link according to the codon present on DNA, so that specific proteins are produced for specific purpose in our body, and thereby DNA control structure and functions of our body. So protein is a biomacromolecule produced and controlled by DNA. So we have our specific characteristic. There are different kinds of species exist in the world with different features, is because of different types of protein encoded by DNA. Now, how these amino acids combine each other to form the peptide bond, that is during translation, decoding message in presence of peptidyl synthesis, that is another part, and we shall learn in 12th class as a part of that synthesis of protein. But here now we are concerned with biomolecules, therefore we have to learn about structure and functions. So in that structure, since thousands of amino acids combine each other, that is how they combine and form the peptide bond, let us know. This is a carboxyl group and this is amino group. Here in this amino acid, amino group, carboxyl group, another amino acid with amino group, carboxyl group. Now, carboxyl group of one amino acid combined with amino group of another amino acid. Here it is shown how it is combined. That carboxyl group can be written as C double bond OH, and NH2 can be written as NHH. So here this HOH, that means H2O, as I told you, that polymer are always formed by the dehydration. So water molecule get released at every bond of peptide formed between two amino acids. See here, this H2O get released and form a bond C double bond ONH. CONH is a peptide bond. Once again, carboxyl group of this amino acid combined with amino group of this amino acid with the loss of water and form the bond called peptide bond. It is clearly with this three diagram and clearly visible to you. So you can understand what is peptide bond and how it is formed. So once again, as a part of revision, carboxyl group of this amino acid combined with amino group of this amino acid with the loss of water form peptide bond, and that is C double bond ONH. So it is very clear, and hence protein are polymer of amino acid. Thousands of amino acid combined to form a long chain of protein. Again another part, that this side, left hand side, this amino group is free, and this, suppose for your left hand side and right hand side, for this is right hand side, that is left hand side. So this side when amino group is free, that is called N terminal, and from that side when all the peptide bonds are formed, the carboxyl group will be free, that is called C terminal. So remember N terminal and C terminal of that polypeptide chain. Now here I have mentioned, no doubt you got it, what is peptide bond. Proteins are polymer, but they are heteropolymer. Why they are heteropolymer? Because of they are made up of 20 different types of amino acid. Starch and cellulose are homopolymer because they are made up of only one type of glucose. But proteins and DNA are heteropolymer because they are made up of different types of monomer. Proteins are made up of 20 different types of amino acid. DNA is made up of 4 different types of nucleotide. And hence protein and DNA are heteropolymer. Starch, cellulose are homopolymer. Now as a part of protein, Therefore, come to the point that proteins are heteropolymer because they are made up of 20 different types of amino acids. Which two amino acids are not take part in protein synthesis, that already we learned on the heading of amino acid, L-alanine and ornithine. They do not take part in the protein synthesis. Now, how peptide bond form, that also I did explain to you. So, come to the next page. As there are different kinds of protein on the basis of structure. On the basis of functions, again there are many types of protein. But we have to make it as a fraction, that on the basis of structure and on the basis of function. On the basis of structure, that there are four major types: primary protein, secondary protein, tertiary protein and quaternary protein. So why they are so called then? Primary protein, they are long simple chain protein, that amino acid in a linear fashion join with each other by peptide bond. So this is a polypeptide chain, single chain, linear chain. For example, tubulin protein, actin and myosin protein. They are single chain protein and hence they are primary protein, and I have given here examples also: actin, myosin, tubulin protein. But when that peptide chain get twist or get arranged in a helical manner, so it is because of that two amino acids get combined by hydrogen bond in between to give that twist to the chain, and hence it is a secondary protein. It is found in nail and hair. This is a helical protein because of hydrogen bond among the amino acids linked in that chain and give this structure. Now, what is tertiary protein? It is again that same polypeptide wound around itself like a hollow woolen ball, but leaving gap and producing some crevices or pockets for joining or fixing of substrate, so as to catalyze the process and produce the product. So tertiary proteins are mostly enzymes. They have 3D structure having substrate binding site, substrate recognition site and ATP binding site, and hence they have 3D structure. They are functional. Once again, a single chain can wound around like a hollow woolen ball, giving some crevices for the attachment of substrate, hence catalytic activity. So tertiary proteins are mostly enzymes and they are biocatalysts, and hence they are mentioned clearly that they are enzymes. Now, quaternary protein is a special protein. This is very unique protein. It is made up of 4 polypeptides, 2 alpha and 2 beta polypeptides. These 2 alpha and 2 beta polypeptides arranged in such a way that they give cube-like structure and their arrangement is specific. Therefore, these proteins are also called as proteins of architecture. And remember, this point can be asked in NEET, that one of the following protein is a protein or biomolecules of architecture or architecture biomolecule. So it is written in textbooks also clearly, but sometimes that simple sentence, single word, that we do not follow and therefore we miss 4 mark in NEET. So please remember this word architecture. Quaternary protein, hemoglobin, is an architecture of protein world. That's all. Now, these 2 alpha and 2 beta chain, they work together, they combine together, and they have very important function to carry the oxygen. If any one of these chain is not there, that molecule is not functional, and that is what then you know that this is what very important. If oxygen is not carried, definitely there will be anemic condition. So these 4 chains are together. If they are missing, then abnormality that to carry the oxygen will be there. So that is important. So for quaternary protein, hemoglobin is the only one protein. Yes. Thalassemia minor, that disease you would have heard, it is because of either of loss of alpha or beta chain and losing the carrying capacity of oxygen, and can create the thalassemia if both the genes are recessive. Yes, okay. Now next, as again part of that continue to be about hemoglobin, which I mentioned, you will get note for here, but on the basis of functions, how many types of protein would be there? There are many types of protein, because protein is such a miraculous biomolecule which control many functions in the living being. And major functions, on the basis of some of that major function, that contractile protein that help in contraction and relaxation of muscles, they are actin and myosin, they are called contractile protein. Immunity protein, globulin protein, immunoglobulin, they provide immunity. Hormonal protein like insulin, many hormones are there, they are protein in nature. Structural protein collagen, that collagen fiber which bind muscles to the bone, tendon are made up of collagen fiber. Transport protein, as already we learned, hemoglobin that transfer carbon dioxide and oxygen. Catalytical protein, enzymes, all enzymes are catalytical protein. Storage protein, myoglobin, store oxygen, and hence on the basis of function there are many protein, many more are there. Now to conclude, naturally you can visit the NEET and see whatever I have explained. Simple protein, that is primary protein with a long chain having that twist because of hydrogen bond. Secondary protein, that same chain wound around like a hollow woolen bond, giving rise to some 3D structure with catalytical activity. So all the enzymes are tertiary protein, and 2 alpha and 2 beta chain linked together to give the architecture of protein called hemoglobin is a quaternary protein. I hope you got it and you understood complete structure of protein and types of protein on the basis of which they are being recognized. Even if you are in doubt, you can ask in comment section. I appeal you to like and subscribe our channel. Also press bell icon so as to receive notification as and when new videos are uploaded. Thank you. Enjoy learning.